Hi everyone, and welcome to the lecture on phylogeny, which I affectionately refer to as the lecture where I struggle to pronounce phylogeny for probably about 45 minutes. Appreciate your patience as I mess up that word multiple times today, but we'll get through it together. And I recognize that many of you might not know what phylogeny is. And it really boils down to a couple of principles that we've already covered in this class and that you've covered in some of your previous classes as well, related to evolutionary relationships and natural selection. So this quote from Charles Darwin really sets the stage for what we'll be learning about today. I would read it to you, but I have tried to record this video 3 times now, and I've messed up this quote every single time, so I'm not going to do that again. I will give you the gist of it though. The gist is that natural selection is a thing that occurs because we have more individuals in every species being born that could possibly survive. And so when we say individuals, that could mean like a single bug, a single dog, a single moose, a single human, you name it, just one entity of that species. There's more that could possibly survive on Earth today. And because of that, there's just this recurring struggle that happens for what will live and what won't live, or what will pass on its genetic information and what won't pass on its genetic information. And Charles Darwin had the idea that if a certain individual in that species had some sort of genetic benefit or evolutionary benefit to itself, so it was easier for that organism to survive because it could get food easier, or it could hide from predators easier, or whatever that might be, it will have a greater chance of passing along its genetic material, resulting in it being naturally selected. And so we'll see that relationship of that genetic material being passed down, and we can use that to build up these phylogenies, which show how all of these organisms and species relate to each other when we're looking at genetics, which is really cool. Again, you know I'm a nerd at this point. I don't have to apologize for being a nerd anymore. So this all boils down to evolution, which is a process of mutation with selection. So the idea that we all generated, all the organisms on planet Earth came from one single common ancestor, and then there was different mutation events that occurred, leading to all of these different speciation events that we see to give us the diverse set of organisms that we see on Earth today. And we know there's a bunch of processes that might cause changes to occur. These can be things like just random mutations that happen, as well as things like genetic drift. So I like to think about genetic drift as like when a small population, say, migrated over to a new area, that small population is likely going to have less genetic variation than the original population, larger population that it came from. I'll post some other resources on genetic drift in the Canvas page, but I'm sure you've learned about them more in your other courses, so I won't go on and on about it. There's also things like reproduction. So for instance, with humans, when you need 2 individuals to come together in order to make another individual, that new individual has that combination of genetic material from both of those people that were involved. And there can also be environmental factors that affect our genetic material. So that's just a short list of them. There's other things also that we could throw in there, but that just gives us a nice taste of what could be going on to drive this evolutionary process. So molecular evolution, which is what we'll be focusing on in this course, is really highlighting the changes that are happening in genes and proteins. And we do this by looking at those sequences. So now that we can sequence that genetic information, we're actually able to look at it and study based on the changes we see, say from one species to another or within individuals in a population, what that potential phylogeny or those genetic relationships actually look like. And that's what we'll be learning about today. So the phylogeny itself is just the prediction of evolutionary relationships. And we have to highlight the word prediction because, again, we weren't there. We don't know exactly what happened, but we can make some pretty good evolutionary guesses based on what we see in the genetic material and in the proteins themselves. Because again, it goes back to that quote from Darwin that we wouldn't see, say, changes in DNA sequences that weren't beneficial for that organism's survival being passed down long term. Those would likely die off when that organisms were created with those genetic mutations that weren't ideal and would not get passed on forward. Oftentimes we represent phylogenetic predictions as something similar to a family tree, and we're actually going to see how we can build up that tree in this lecture today. So we have in all of science this thing called the phylogenetic tree of life. And so that goes back to the very common ancestor that we all share, which would be down here. It refers to us all coming from a common origin. Same with all of the organisms that live out in the world today. And then we have these different branches that come off of that for different subspecies or groups of species. So you can see here we have all the bacteria over in one group, all the archaea over in another group, and all the eukaryota over in another group. And we could break this down more and more. It would just get more cluttered on the slide. So we have this at a fairly high level. Predictions within phylogeny involve extinct species. So when I say we weren't there, we don't know what happened. We weren't here when the dinosaurs were roaming the earth. We don't know exactly what happened. We don't know how those dinosaurs were related to other things. And this makes it a much more difficult problem. It would be impossible for us to know all of the actual events that happened to build up this tree of life. So what we do is we generate inferred trees instead, based on numbers and statistics that give us our best guess into the evolutionary relationship that existed. So the goals with phylogeny are to determine the relationships between different species, but also to determine the time of divergence. So by looking at our sequences, we can actually estimate how long ago a specific divergence occurred between two different species, which is really cool. Any set of homologous sequences can be represented as a phylogenetic tree. And we've seen this when we've built up multiple sequence alignments. You can see those guide trees that we had, and it showed potential evolutionary relationships. As long as we have some level of confidence that we are using homologous sequences, I told you I was going to mess up the pronunciations of things in this, we can build out a phylogenetic tree on that. So unlike sequence alignments, where we just had that guide tree that helped us identify conserved regions, a phylogenetic tree, it will actually help us infer relationships between things. So with the multiple sequence alignments, we would say, okay, yeah, these things are likely related because they share this conserved region of, say, an amino acid sequence. That would mean they have a similar protein domain or something like that. But we wouldn't know which one came first or which one was more related or less related or what that actual evolutionary relationship looked like unless we did a phylogenetic tree. So you might be wondering, how in the heck do we actually measure or calculate how long a divergence event occurred? We have this thing called the molecular clock hypothesis that states for any given gene or protein, the rate of molecular evolution is approximately constant. And this was discovered back in 1962 and then tested in 1971 by a scientist named Dickerson for 3 different proteins. And what Dickerson found is that each of these proteins had a different rate of change. So if you look over here at this graph, you can see we have the cytochrome C protein here, we have the hemoglobin protein here, and we have the, I don't even know, I can't read that, something peptide over here. So 3 different ones. Along our x-axis, we have the millions of years since divergence. And along our y-axis, we have the number of corrected amino acid changes per 100 residues. So the reality is these were all different rates. So our rates would be our slopes that our different lines are at. So these aren't all overlapping each other. They have lots of space in between them. but they were all linear. So we have these different time points on these lines that represent different sequences we have, and they fall in a linear or a straight line all the way down to that x-axis. So that's why we say that it will depend on the protein or the species or the gene that we're looking at, but if we know that rate or that slope, we can calculate the time since divergence. And how we actually do it is this formula here. So we have R, which is our rate, K, which is our number of substitutions per site, 2, which represents that we're comparing two sequences, and then T, which is the time of divergence between those two sequences. If we wanted to solve for T, we just have to rearrange this formula. So how we do that is we multiply each side by 2T to get it out of the bottom. So then we would get 2TR equals K. And then we would divide each side here by 2R to get T equals K divided by 2R. Just a quick little math refresher there. So that is how we would rearrange it to calculate the time since divergence, but sometimes we also do want to calculate the rate of change if we already know the time since divergence. So I already kind of went over this, showed you the reorganized equation for solving for time, mentioned that we need that actual rate in order to solve for that time. So if we had, say, the same protein in a different species, and we knew the rate of change for that protein, we could infer that that rate might be, or is very likely, very similar for a different species. So we could estimate that time of divergence for the new species to others. This isn't exact science. There's a lot of should be okay, looks like it'll work, mystical, magical hand waving. It's not that bad. But it is an estimate. It is not an exact thing. It's not saying this happened exactly 5,000,008 years ago in 2 days. That's not the level of accuracy we're looking for here. It's a general kind of evolutionary guess at when this might have happened. And we are working on the scale of like millions of years. So when we think back to like our significant digits, if we have, say, a rate that has 3 significant digits in it, our millions of years to be like 3 significant digits is still only significant within like the thousands, 100,000s of years. So, it is an estimate, which is really important to remember. And you'll see why that's important to remember in a future demo that I have in this lecture. We can also solve for R. So R uses just the same equation that was originally shown without all of the substitutions. So say we have our time based on, our time down here, based on fossil data, which we can get time based on fossil data or carbon dating. And we can calculate the rate for that. If we know, say, the specific protein, how many substitutions per site occurred. So that is typically looking at every single position, how many substitutes per site occurred. And so we can solve for all of this to get our overall rate. There's a lot of terminology that we use in phylogeny. We'll go through a little bit of it here. So a synonymous mutation is a mutation in our DNA that results in the same codon, which would typically be in position 3 of a codon. So if you remember back to the amino acid table, you'll know that there's lots of different codons that can code for the exact same amino acid. And so here we would show different options of synonymous mutations. So say for leucine, we could have CUU, CUC, CUA, or CUG. And if we have any of those nucleotides in the 3rd position of that codon, it would result still in a leucine being in that position of the of the protein. And so that would be what we call a synonymous mutation. A non-synonymous mutation is a mutation that would result in a different codon. So I challenge you to take a look at the amino acid or the codon table and find a couple examples in there of amino acids that if we don't have that specific nucleotide in the third position would result in a non-synonymous mutation. Throw it up on the discussion board and we'll see who can find it first. That'll be awesome. So, as I promised, there was going to be a little bit of a demo here. So there is a way or a site that we can use to predict the divergence time. This is called tree time, and I'm just going to open it up here and then move over to it. 2 seconds. And it looks like this. So Tree Time of Life has a whole bunch of information on it, all based on public information that has typically been published in peer-reviewed research articles, and then gives us these searchable trees that we can investigate. Also shows things like atmospheric pressure and oxidation over the years. Doesn't mean as much to me because I never got real deep into evolutionary science, but for the people that are real deep in there, means quite a bit. So if we wanted to get the divergence time, we could use this very first search here and say we wanted to pick any other taxon. So I like to let's pick cat and dog. Why not? I'm not even putting the scientific names in. It will figure that out for me, which is beautiful. So you'll see over here, it's resolving my ambiguity. It picked out my scientific names just based on those general ones that I predicted. And so if we scroll down here, so this is what I'm talking about with this wild graph. So it has geo timescales, Earth impact, solar luminosity, CO2 levels, O2 levels. They just don't mean anything. I don't know. They mean something. That was the worst thing I could have possibly said. They don't mean anything to my brain right now. What I'm looking at here is this million of years ago, MYA, million years ago, timeline that we get from it. And so if we look over here, the median time since divergence is 55 million years ago. And we also have an adjusted time down here. And if we scroll down, we see all of the different papers. There's multiple pages that suggest the times of divergence for these two species. And you can see over here on the time side, they have lots of different values. As I mentioned, it is an estimate. There are lots of different ways we're trying to make these things more accurate. And so you can see as different years go on, we're getting different values. Sometimes even within the same year, we're getting different values. And so this one here, the median time, is a median, which is similar to an average, but it's just the most frequent value. The median time that was reported across all of those different papers. So instead of having to go and look for them, you can just find it right there, which is really, really convenient. If I go back to the search tab, there are some other things that we can see as well that you can play around with. So you can get an evolutionary timeline for a specific taxon. So if you wanted the evolutionary timeline for dogs, you could see exactly when those diverge and what all those historical ancestors were. You can also build a time tree for a specific group of taxa if you wanted to do that, or uploading a custom list of species. I won't ask you to do any of those things in this class, but they are available should you be interested. All right, just flipping back to the slides here. So as I mentioned, I think this might be like my 5th time mentioning it, but it is important. Molecular calculations are an estimate of the time since sequences diverged. Rates are organism and sometimes protein dependent, so use them with a grain of salt. And the calculation is not perfect. So we don't always know how many times a substitution or a mutation occurred. For instance, we could have a situation where we had an individual nucleotide, say it was an A, it got mutated to a T, and then it mutated back to an A. If we don't have species or sequences that represent each of these time points, we don't know that all of those things occurred. There are ways that we can build in some adjusted sequence counts or mutation counts, but it is a very tricky thing, which is why they are estimates, which is why you see all of those different numbers on the tree of life, and why we use that median from the tree of life or from other sources in order to infer when those divergence evolutionary relationships happened. As I mentioned previously, phylogeny is the prediction of evolutionary relationships. And so that is a prediction, our best guess based on our hypothesis that we currently have. It is not a statement of truth. It is a very reasonable, educated guess, which is important to know. I also mentioned that we will build out trees that are similar to family trees. So you might be wondering, what the heck are all these tree things? Don't worry, I got you covered in this next section here. Of course, new type of thing we're learning about in this class, new type of language that we need to learn all around it. So trees would be like this whole collection that we have here, this whole symbol. If we were thinking about this in terms of a family tree, we would have things like parent 1, parent 2, child 1, child 2, etc., etc. Ancestor, which would be maybe like a grandparent of some sort 1, and it could go back and back and back, all sorts like that. I'm going to get rid of all of that little beautiful annotation I just did so that when I start talking about these other things on here, it doesn't get too confusing. Typology is the first new term that you're probably learning in this class related to trees, and it is simply just the layout of the trees. So it starts up at the top, it's a rooted tree, you'll learn what that is in a bit. It has 3 leaf nodes, which are the ones that are at the very bottom and have no further children. It has this clade over here, those kinds of things. That is a topology. So it's just about how the tree is actually laid out. All trees are what we would call in computer science directed graphs, which means we have one way of traversing or going through the tree. It always has a directionality associated with it. And in the case of trees, we're always moving downwards. So we can only move down that branch or edge. We can't move back up it. So once we're there, we can't go back up. And similarly for this, we can only move down. And so you'll see we have these arrows at the bottom that indicates the direction of those branches or edges. Every line that is within a tree is called a branch. So these ones down here, these are branches. These here, branches. Also called edges because that's what we would refer to them in computer science. Nodes are the dots. Dots, 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 dots. In bioinformatics, these are typically going to represent an individual sequence or a hypothetical common ancestor, something like that. Nodes can also be called leaves if they're on the very bottom of the tree, so they have no further branches coming out of them, or they can be called taxa because they typically represent taxa in bioinformatics. With trees, we use parent and child and ancestor terminology typically. So for this node here, it would be considered a parent of these 2 nodes down here, but not this node because it's not connected through those directed branches. So this would be the parent node to these 2 children nodes down here. But this node here would also be a child node to this parent node up here. So it always has that, like, depending on what level you're looking at, the definition could switch. A common thing that we'll do in, say, exams and things like that is, given a specific tree, if I tell you, say this was called node B, I would say, what is the parent of node B? And say this was labeled node A, we say node A. And what were the children of node B? Say this was C, D, and E. We would, oh, sorry, I put those on the branches instead of the nodes. They should be with those down here. We would say the children of node B were D and E. So those kinds of questions will come up often in exams. There are different kinds of trees that we have. We can have rooted trees or unrooted trees. A rooted tree means that we have a special node in our tree that doesn't have any parents. So that would be an example of this root or this node right here. And then every other node in the tree has exactly 1 parent. So if we look up from any leaf node, we always only have 1 parent. There might be other ancestors that are further up, but we don't have something like we would often see with family trees where we would have 2 parents going into 1 node. In phylogeny, it's always only 1 child, or a child will have a single parent node because it is the single common ancestor. Phylogenetic trees are typically rooted trees, so you'll often see them in this kind of topology, where our leaves represent our existing organisms that we are measuring or we have sequences for. And then internodes, which are ones like these ones here, represent extinct or hypothetical organisms that would be common ancestors that we don't always have the sequences for. A clade is what we would call a subtree within a tree. So if you pick a node within a tree and you look at all of the things below it, everything, including that node that you are currently looking at, would be considered a clade. We could also pick this node, and everything that was underneath it, we would consider that to be a clade. That would just be the whole tree in this case. Same with this leaf node here for fly. We'll just have a very small clade over here. So effectively, it's just saying, pick a node, everything under it, that would be what we would call a clade. Within phylogenetic trees, we have both scaled and unscaled trees. So scaled trees are when our branch lengths are proportional to the differences between our sequences for neighboring nodes. An unscaled tree, the branching is just representing the relationships, but it has nothing to do with those differences or the distances between neighbors. And so this gives us an example here. So we can see here, up in this top box, we have rooted trees with scaled branches. Often we know that they're scaled branches because they don't line up nicely, like the description of what they are. They're all at different spots within that tree, kind of all over the place, but it's because these different branch lengths need to be different lengths to represent those differences. When we have an unscaled rooted tree, which we see in this second box here, you'll see that all of those species names line up along the leaf nodes, that we still have different clades within a tree that we can see. So here's some examples of clades, but they're all lining up. So we're still seeing those relationships. We're just not seeing the branch length directly related to how different those sequences are. We can also have unrooted trees, not very common in phylogeny. That just means they don't have that hypothetical ancestor. So you see them at the bottom rectangle here. They look kind of like a spider web is the easiest way to spot them. You can see they have things all over the place. They're harder to identify scaled and unscaled. So the scaled ones, you'll have to really look closely to see, oh yeah, these are different lengths. The unscaled ones, they're all the same length, but a lot harder than when we have a root and we can just say, oh yeah, those are all aligned down one thing. We won't see these commonly in phylogeny. Of course, we have different ways of representing these trees. So when we're working with a computer, if we were to pass it this image, especially when we first started doing phylogeny as bioinformaticians, the computer wouldn't know what to do with this. It would be like, what in the heck? So we came up a way of communicating with the computers about different tree structures. And how we do that is through these Newick representations. So this first one here is a Newick representation of this tree here. And it is unscaled. So all the branches are lining up at those leaf nodes down there. And what you need to do in order to represent or figure out a Newick tree representation is you just have to identify the clades. So for instance, we have two clades here when we look at this level, one with B and C and one with D and E. And all you need to do for the Newick representation is put those in brackets. So you'll see here we have B and C in a bracket, and then we have D and E in a bracket. And then we move up a level, and we see at this node here, we have a clade that contains A, as well as that subclade B and C. So we put that whole thing in brackets. So you'll see the B and C bracket is now contained with an extra bracket on the outside, and then we add comma A with another bracket. And then we move up to the final node and we do the same thing. Okay, now we have that ABC clade and that DE clade when we put the whole thing within a bracket. And so it's just kind of nesting these brackets up, starting at our leaf node clades and then working up one level every week. If we wanted to add a scaled tree representation of a Newick, all we do is the same idea. So we take, again, those clades that are starting at the beginning. So we have one here for B and C, one here for D and E, and we just add this colon 1 or colon 2 or colon whatever the branch length is after the label. And that will help us figure out, okay, so this was a 2, this was a 1, etc., etc. We can rebuild out all of those branch lengths. A little bit more complicated to figure that out because we do need to do some math. So when we get into things like, say this A is a 2, but then we also have this BC clade and we have to figure out what this is, we can do that. We just have to do a little bit of math in order to get there, which makes it a little bit more difficult. You'll see these kinds of Newick representations if you take a look at the multiple sequence alignments and the guide trees that are generated. Oftentimes they will give us those guide trees in those Newick representation formats so that we could paste those in to say like a tree viewing software or something like that. And then it would generate these beautiful images that I've marked up for us, which are much easier for humans to digest. When it comes to building trees, there's 3 general methods that we'll be going over. So we have distance-based methods, parsimony-based methods, and probabilistic methods. So starting out with our distance-based methods, trees are constructed by determining pairwise distances between sequences. So for us in this class, this will be like number of mutations. The starting sequences have to be homologous for this to be accurate. So again, we can just throw sequences to the computer. We can say, do something, make me a beautiful tree with it, and it will go and dutifully do its job and bring us back a tree. But if they aren't homologous sequences, that resulting tree won't be accurate whatsoever. So we always have to have our biology hat on, thinking, are the things that I'm inputting to this software going to make reasonable biological sense? A very common method under the distance-based methods is called UPGMA, which stands for this very long, well, it's very accurate, I guess. It's long, but it's accurate description of what we're doing. Unweighted pair group method with a arithmetic mean. Like, so exciting, right? But the general strategy for this is these 4 steps. So we create a distance matrix based on the similarities or differences between pairwise sequences. So we're going to be counting mismatches or different things like that, similarly to what we would do for like a multiple sequence alignment to figure out, okay, between these 2 sequence, this is the number different. Between these other 2 sequence, this is the number different. Once we have that distance matrix, we take the most similar pairs and merge them into a single taxon or a clade. We then recompute the distance matrix based on that clade and repeat these 2 steps until we have fully constructed the tree. As we move up the tree, the smallest distance might result in merging 2 leaf taxas, merging a clade with a leaf, or merging 2 clades itself. And we'll see examples of this here. So I know talking about that is fine and dandy, but let's actually try to do something with it. So in this example, we are going to construct a phylogenetic tree from 3 sequences that are from different species. These species names, for the sake of argument, are just A, B, and C. They could be cat, dog, and parrot. I don't know. Pick something if that helps. And we are given our initial starting distance matrix that we have. So we have each of our different sequences, and then we have the number, say, of, let's call this mutations between the different pairwise sequences. So from A to B, there was 1 mutation. From A to C, there was 3 mutations. And from B to C, there were 2 mutations. And so that gives us this distance matrix. We don't need to worry about everything on this triangle because it would just be repeated. And so what we need to do is we need to figure out, okay, well, which one should we be starting with by looking at this distance matrix here? And if we remember, our first step is pick the ones that are most similar. So if we're saying these are the number of differences, the ones that are most similar are going to be the lowest number. And in that case, it's going to be A and B. So what we need to do is make those 2 things a clade then. So we merge those 2 leaf taxa together. So we start off with our tree here, and I'm going to show you how we would type these things out, because you will be asked to type these things out for things like assignments and stuff in text files. you just have to kind of mess around with your text file to make it look somewhat reasonable like a tree. So you see how I've done that here? I've used dashes and vertical lines to represent a taxa that we have here. So that's going to be our first tree that we have. And then we need to recompute our distance matrix. So how we do this is we now have a distance matrix that's going to look something like AB, because that's our taxa, and then our remaining leaf node is C. And we'll do that again down the side, and I'll just add some spaces here to make sure everything is spaced out nicely. And then I add my C down at the end here, give it some more spaces so it looks nice. And again, we don't need to worry about things along the diagonal or in the top triangle, because those are just going to be repeated by what we find down here. And so down here, we're looking at the distance between the AB clade that we'd selected here and C. And how we figure that out is we do the distance between A and C plus the distance between B and C, divided that by 2. And in this case, let me just put some square brackets along this to show our order of operations that we'll have. And so if we go back to our original, we see our distance between A and C. If we go back here, we go to A, we look for C, that is 3. So we're going to sub 3 in there. So 3 plus our distance between B and C is 2 in our original matrix, and then divided by 2. That is going to be 5 divided by 2 when we simplify that, which should equal a value of 2.5. And so what we do is we go back to our recalculated matrix and we add that value in, 2.5, to that matrix. Oh, I got to tab some things over here so that it lines up nice. There we go. And now we repeat. We start adding in our next section to our tree. Oops, pressing the wrong copy paste. Copy. I would like to copy this whole thing. Oh, it won't let me. Okay, well, whatever. I'll copy the text and add it in then instead. Oh, Adobe Acrobat not working the way I would like it to. So this is what we had so far. Our next smallest or most similar are going to be the AB clade that we had, with C. So that would be that 2.5 value. So we need to add that in then to our tree. And to do this in a text file, you just have to add spaces as you go. I've done this a couple times, so I kind of know-ish how many spaces I need, and I'll still mess it up, and that's okay. And down one, give me my vertical, and then give me a new line with this, and I have my C. And so you'll see that I have lined all of these up along the edge so that they're roughly kind of in order there. I should add in another dash here, for my root, and just space everything over 1, maybe 2. Give it a little more space here. There we go. So you see, it's not perfect, but it's pretty close. It's good enough for an assignment when you're working in a text file and you're not a computer. And so you'll see I have my clade of AB here, and then my C that was added on to the end. There's no other things that I can add into this distance matrix, so we know we are done. This here is an unscaled tree, but we could try to make it scaled. So what we would have to do in that case to make it scaled, of course, I keep on hitting the wrong copy paste, which is half my problem here. Oh my goodness. Control C. Control V, there we go. To make it scaled is we just have to go back to those distance matrices, and then along the branches where we're merging things together, we just take that original distance and divide it by 2 to get the branch length. So for instance, for our first clade that we made with A and B, our distance was 1, so our branch length up here would actually just be 0.5. And one way that we often represent this in text files is just by putting that number there. 0.5. You don't have to do it for both. You can just do it for one. That's perfectly fine. Our next one, which is our distance between AB and C, was 2.5 5. So that should be, if we divide that by 2 again, 1.25, which would be our distance that we have on this branch underneath C here, which is 1.25. The only remaining branch we need to figure out is this little branch here. And how we get that is we do 1.25, which is the full distance here, minus the 0.5. Because if we go from C and we want to go to A, or we go from C and we want to go to B, we should only be taking a max distance of 2.5 to get there. So we already have the 1.25, we already have the 0.5, we just need the rest of that piece there, which I'm gonna run out of space typing this, but that's okay, would be 0.75 if my mental math is working correctly. And now I will tab everything over a little bit more to give some better spacing here, so it's not all mucky. And we don't have a bunch of things that look just a mess. And you can also try to mess with like the number of tabs that you have, different things like that, or dashes, sorry, to try to make that a bit more of a representative. But as long as you are including those numbers, we know that it is a scaled tree. And that's how you get a scaled tree out of an unscaled tree using those distance matrices. We have a lot bigger of an example, which I would really like for you to try by yourself to see, hey, can I get this working for these larger examples? If you are struggling, if I get enough votes on Canvas, I will post a video going through this example. But what you need to do for this one is you need to do pairwise comparisons of each of these sequences to count the number of differences between them to build up that distance matrix. Why don't we do that first together, and then you'll have that as a starting point, and then you can use that method where you find the smallest, you recompute, build the tree, find the smallest, recompute, build the tree, yada yada, over and over until you get that final tree built out. So we have 4 sequences in this case, A, B, C, and D. I'm just going to add in my placeholders for them. And then I'm also gonna dash out that first line because it will all just be repeated on these other ones. So the very first one that we need to start off with here is A versus B. So if we look through this, what we need to identify is the number of mismatches effectively. So we have ATG, ATG, we have one mismatch here, we have one mismatch here, so that brings us up to 2. Here's another one, 3, 4, 5, 6, 7, and that should be it, I believe. So that is 7. And if I make a mistake and I give you the wrong distance matrix, I apologize. It's hard for me to do these things with just looking on the fly. And not having like a highlighter or something. Actually, I think I do have a highlight I can use. Perfect. Okay, so now let's go to A and C for our next one. So you'll see we put A and B by just going down the A column, and then we did that B row. So for A and C, we have A, T, G, G, C, C. Here is a mismatch. A, G, G, T, 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 T. Here's another mismatch. And another one. I swear I'm going to miss some. I'm very concerned about me doing this and not having a computer do this. And, oh yeah, almost missed that one. So that's 1, 2, 3, 4, 5 that we get from that. And we can just add that in here. 5. Now we can either keep going through the A's or we could fill out our C row. It doesn't really matter. I am going to continue to go through the A's because that works for my brain. But if you wanted to finish the row instead, you could as well. I'm just going to delete all of these highlights so that I am not confused when I look at this again. And now we're doing A versus D. So A, G, here is 1, here is another, here is another 2, G, T, T, here is another, GGCG, here is another. Again, I'm just doing a pairwise comparison with my eyes here, trying to make sure I don't miss any, and I don't think I did. 1, 2, 3, 4, 5, 6, 7, 8, 9. So that will go then here in this spot here. All right, so that whole A column is done. Now we'll move and we'll compare B with C. I'm just gonna, again, get rid of these highlights so we don't get too confused about counting and what's going on and having our eyes be distracted. Exercises like this always remind me why I love computer science and bioinformatics so much, because I don't think I should be trusted to do any kind of manual work like this. And I was trusted to do manual work like this when I was a summer student in a research lab. That was before I knew how to do bioinformatics, and we used to go through multiple sequence alignments, like highlight things in a Word doc. It was wild. All right, so for B and C, my first mismatch I see are these two CCs as well as this A. Going through the rest here looks good. Here's another one. And then those final ones look good. So that should leave us with 4. And that's going to go, so we look at the B column and then we look over at the C row. That's comparing the two of those, and we put a 4 in there. Because the rest is on the diagonal for that row, I'm just going to add my dashes in so that I remember I don't need to look at those. Our next one that we'll compare is B with D. Once I delete all of these highlights, I like that I'm getting less highlights. So B with D, we have ATGG, we have 2 mismatches there, 2 more here, GTT, 1 more here. Because remember, even though this G is the same as that A up there, which my brain almost tricked me into thinking it was the same, we're comparing D with B, which has a C there. And we have a C here, GAs, GG, CGs. Also reminds me of how similar G's and C's look. Wild to me. Just how we got this right at all is just wild. So we have 1, 2, 3, 4, 5, 6, 7 for that. And that is correct. 7. Again, we'll just go through and delete those highlights. And then we're going to do our final comparison, which would be C with D. And then once I give you that distance matrix again, I would like for you to go through this on your own, see if you can get the solution that I'll post yourself. I can do a video walking through it as well, but I think y'all got this. So comparing C with D, we have 1 mismatch here, 1 more here, 2 more here, 1 here, 1 here. GGCs, those GGCs always get me. Another one there, another one there. That's 1, 2, 3, 4, 5, 6, 7, 8. And that is correct. Ooh. I have my solution over on the side here, and I'm proud of myself that my mental checking actually worked. So that should be what our distance matrix is. So remember, what our first step that you'll be going through yourself is looking at the distance matrix, finding which one is the most similar, making a clade out of that one, and then recomputing the distance matrix. So the one that's most similar again is going to be the one with the lowest number of mutations. So that in this case will be C matched with B is where you're going to start from. Best of luck with that. I know you got this. This is the solution that would come out of it in the end, so you know that you got the right solution if you built that out correctly. Again, this is an unweighted or unscaled tree, so it doesn't have any of the branch lengths associated with it. We know that because all of our taxon are lined up along the bottom there. If we wanted to make this scaled, we just go back into that distance matrix. Every time that we're pulling in a new clade or making a new group, we calculate what half of that distance would be and label that on one of the branches, because the full distance would be going all the way from one to the other one. So that's usually going up and down 2 branches. And that was why we have to divide it by 2. So matches and mismatches. I knew I was going to mess up my pronunciation in this lecture. But matches and mismatches are not typically used to calculate the distance matrix in the UPGMA method. We will use them for this course for simplicity, but we often use adjusted values. And so that's going to be things like the Jukes-Cantor model or the Poisson correction model. There are more details on this in Bioinformatics 351 if you're interested in that. If you remember back to before we got into all this tree building madness, I mentioned there was 3 different methods that we can use to build trees. One was the distance method that we just went over, and the next was parsimony. So parsimony is where preference is given to one tree over another based on which one requires the smallest number of changes. This is effectively Occam's razor, if you're familiar with that, like the path of least resistance. The trees are typically going to be unrooted, which is very different to what we typically see with phylogenetic trees. Remember, that's that like spider web tree that's all over. The intuition behind this is that mutations are very rare, and the model with the fewest is likely the one that will be the most correct. So again, Occam's razor, our path of least resistance, how can we get there? That's exactly what parsimony is. And maximum parsimony is the best tree is the one with the shortest branch lengths possible. So not only are mutations important, but also we don't want to put in like these really big branches just to get the shortest number of mutations that would represent really large evolutionary distances. You want to kind of balance those number of mutations with also the time or the distance there that we would see in the trees. So to do maximum parsimony, we construct an optimal sequence alignment. We know how to do that from the previous lectures. And then for each position within that multiple sequence alignment, we construct a tree. And we score the tree based on the number of evolutionary changes needed to produce the observed sequence changes. The tree with the lowest score is the one that we would select at the end. We've talked a lot about different kinds of trees, and with maximum parsimony, now we're comparing trees, but do we need something that's like e-values for trees? So if you remember back to our BLAST lectures, e-values are what allowed us to compare trees along different BLAST searches. So they were a way for us to say, oh, sorry, no, that's bit scores. Wow, you can tell I need coffee. Give me a second. Give me some coffee. E-values told us how significant our search results were based on the database we were searching in and what was the likelihood that we would just get that search by random chance. Do we need something similar for trees? Like, what is the likelihood we would just get this tree by random chance? If you've been following along in this class, you probably know the answer is yes. We love to have confidence in our results because computers will just give us results. And so if we can have some sort of confidence in that's really good. The way we do that with trees is something called bootstrapping. And bootstrapping is how we measure the robustness of the tree's topology. It's a measure of how many times the same topology was observed if we repeated that phylogenetic tree on a resampled set of data. The higher the values, the higher the confidence we're going to have in the tree topology. So that's effectively saying that, okay, if we resampled this data, generated a tree, resampled this data, generated a tree, resampled the data, generated this tree, how often would we get a tree that looked exactly the same as the tree we're currently seeing? Or I guess flip it, because the higher numbers in this case are better, whereas the e-values, the lower numbers were better, but same kind of intuition there. So for phylogenetic analysis, typically what we do is a multiple sequence alignment, followed by some kind of tree building, and then followed by some kind of bootstrapping. The multiple sequence alignment is going to be our first step. Then we get into our tree building. We're using whatever method that we're interested in. And then we use bootstrapping to evaluate how confident we are in our results. There are lots of different phylogeny programs. So they can use ClustalW, you can use Mega, you can use Philip. Feel free to take a look at those, try some out. We'll likely try some out in an assignment in the future. If you want some additional readings on phylogeny, you can look up these chapters in those textbooks in the library. I don't think I messed up phylogeny that much in this lecture, but I definitely mispronounced other things, so you're welcome. Have a great day.